Hello everyone. So in this video, I'll be talking to you about how to use COPILOT for research methods. You'll be aware that COPILOT is actually a GPT-4 based artificial intelligence search tool, which is recently launched after OpenAI came out with ChatGPT 3.5 and 4. So let me just explain you how it works, and primarily I'll be looking at how you can use it for developing research methods. So since I am a medical professional, I've used a life science subject to explain you the concept, but you can use it for any other scientific research. So a brief outline about what you mean by GPT. Now GPT stands for generative pre-trained transformer. So in that, pre-trained is something which is self-explanatory. So it is actually trained with a vast amount of text from a lot of resources like books, articles, websites. So in other words, it's a extensively pre-trained, a large database. You can call it as a huge sort of an internet plus additional material. So based on this, the tool can generate things. So you can see that generative is again a simple explanatory term. So it has the creativity to generate original text as if it looks very true and new and original. Now transformer is something which is not a fully explainable in a simple concept. It is a little complicated uh, machine learning concept. So basically it is an architecture which actually helps the tool to understand the connection between words. Actually it uses what is called predictive text. When you give a particular phrase with say 3 words, it can predict what can be the 4th word and the 5th word. So that's a basic concept by which the entire artificial intelligence, as far as these general purpose artificial intelligence work. So this is something you can look at. You can see a huge database where there are a lot of computers and a kind of a networks which happens in the information. So these are basically what you call as a large language models, which has a lot of information within it, and it is a language based model. So which means that you need to speak to it in a specific way so that you can get the maximum information from the tool. So before I go into what is called a prompt, so you can just remember that you need to log in to the COPILOT using the web link given there. So you can even use it without logging in, but if you log in, the response will be good, and certain few features are given to you only when you log in. So as of now it's free, but if you have a subscription for OpenAI, it will directly access it, and you will be able to store all your search in it. So basically this is something like this. You sit in front of a computer or artificial intelligence, and then you just want to extract what you want to extract, but you need to give the correct command to extract what you actually need. So first let me tell what this mean by a prompt, right? So prompt is nothing but the language or the method of speaking to the artificial intelligence tool. So there are many methods by which prompt can be done. So the why I am calling this as a primary or the mother prompt is because the first prompt is very important, because by default most of these artificial intelligence tools work as if they speak to a 8th to 10th grade individual. But here you need to give a particular instruction to the model so that it gives you the precise information you need to get. So there are four things I would say you should use in the primary or the mother prompt. That's the first prompt you give to the AI tool. So first you need to tell who you are. Second, you need to tell what is your intention, why you have come seeking help to the GPT, what you want the AI model to act as, and what you want the GPT to actually do for you. So I've used this example of SARS-CoV-2 infection which causes COVID, because most of us are aware of it. And I've used mitochondrial injury because we all know what is mitochondria as a 10th grade student who have learned science. So first thing what we are telling is that who you are. So we are telling that I am a cell biology researcher. So you are just telling who you are. So cell biology researcher means that you are a postgraduate and you expect a higher level of information from the artificial intelligence model. Now then you are telling what is your intention. So you are planning a PhD on mitochondrial injury due to SARS-CoV-2 infection. So you are telling your intention. So then you want the GPT to act as your PhD guide. So the level of the information again you are asking from the GPT model is quite high. So then you are asking it what it should do to you. So you are asking it to give you few research questions on the topic and explain the hypothesis. So it will generate few hypotheses and it will also explain you the hypothesis. So this is what I am going to give. I'm going to demonstrate you in the COPILOT tool, online COPILOT tool, and we are going to see how the prompts work and how well we are able to get the information from the GPT-2. Now we are into the COPILOT. So I've actually logged in. You can see my name in the top corner. Now what I've done here is that I've just put the prompt which I have already explained in the initial introduction. So you can see here we have put I am a cell biology researcher. So we have mentioned who I am. So next, that's the first prompt, who I am. So second thing what we have done is, so what is my intention is to plan a PhD on mitochondrial injury due to SARS-CoV-2. So that is my intention. Then I put what the AI tool has to act. So can you act as my PhD guide? And then the last one is to give a research questions on the topic and explain the hypothesis. So fourth thing we have given. So you can see down what information it has given you. So it is replying that certainly it's willing to act as my virtual PhD guide and it is willing to tell me about the mitochondrial injury caused by SARS-CoV-2 infection. Now it has given three research questions. First thing what we have given is, how does SARS-CoV-2 directly impact mitochondrial function? So for that it is telling me that you have to investigate the virus interaction with specific mitochondrial components like electron transport chain, oxidative phosphorylation, right, and mitochondrial DNA. And it is also telling me to see whether the viral proteins can interfere with the mitochondrial process and make it dysfunctional. So the next research question it is telling, what is the consequence of mitochondrial damage during SARS-CoV-2 infection? So it is telling what happens after the infection, how does the mitochondrial function affect the energetics of the cell and the overall health. The third one, it is giving me whether SARS-CoV-2 alters mitochondrial dynamics. For that, it is asking me to look into the fission and the fusion dynamics upon viral infection. So when I go through this, and below that it is also given the hypothesis, mitochondrial injury hypothesis, supporting evidence it has given. So it has also given certain references for whatever. Some are just newspaper references, but these references are not the purpose where we are looking at. These are basically, though it has come in newspaper, it would have been from a scientific article. So here you can see that it is giving link to a scientific article. So anyway, so it has given us 3 research questions. So I feel that I have decided to choose the first one because it is looking at the electron transport chain and oxidative phosphorylation, which I feel which can be looked at the lab. The second one is more like I need to look at the patient follow. So I have to take up infected patients with COVID and I have to follow up and see them. And fission and fusion dynamics are quite advanced concept. Okay, so I am quite happy with the first response. In case if you are not happy with the first response, you can always ask to regenerate the response. Okay, so you can request it to, you can tell that it is not that good and you can always try to regenerate the response. Right. Now, so I am happy with that. So the second prompt, so based on the mother prompt, I've got a reasonable output from COPILOT. So I'm just thanking you for the suggestion. So always thank the AI model because it is a kind of encouragement you give to the model. And people say that when you appreciate it, it tends to get more information, which is in line with what it has already given. So what I am asking now is that, so since I already told that we have chosen the electron transport chain, so I am asking to suggest laboratory method to assess the function of the electron transport chain. So I have decided that I will look at electron transport chain and also the oxidative phosphorylation. So here I am giving a straight prompt because the mother prompt is already there. So this is the same thread. So when you search in the same thread. So can you suggest laboratory method, because I am a laboratory scientist, to assess the function of the electron transport chain. And I'm also asking it to suggest what sample should I use, because I need to take samples from the patient. So is it blood or saliva or sputum? So 3 common samples which you can get from people who are infected with COVID-19. So for this it is responding, telling that for assessing the electron transport chain, it is giving the methods. So it is telling about the spectrophotometric assays, and it is also telling about what these assays actually do, and it is also telling the sample. So because in the prompt itself I asked it 2 things, one is the method and the sample. So it is telling that you can use tissue samples and cultured cells. So then here it is telling that for here you can look at, second is the extracellular flux assay. It is telling liver sample cells which are difficult to get. Mitochondrial respirometry it is suggesting. The method it is telling, and it is asking for the fresh isolated mitochondria or intact cells. Okay, so for the ETC it is telling. So then again it is coming down about suitable sample types. One is tissue biopsy. Another is telling peripheral blood mononuclear cells. So it looks impressive for me because I can get a blood sample from the patients infected with the COVID-19. It is also telling cell lines, live cells, which may be difficult to get. Platelets, another things it is telling. So platelet rich plasma from the whole blood. So I can take the blood, I can take the platelet rich plasma and see. So it is giving me some option for the samples also. So again the response is impressive. It has given me what tests I can do for electron transport chain, and it is also telling me what samples I can take to assess that. So since the oxidative phosphorylation was my next question, I put that as the next prompt. So here I am asking the same thing, laboratory test for oxidative phosphorylation. Again, what sample and what to perform those tests. So for that it is again coming up with the reply telling that you can Study the oxidative phosphorylation with high resolution respirometry, spectrophotometric assays, and extracellular flux analysis. It's given me the methods. Then it has given me the types. So tissue from skeletal muscles or liver, which is difficult. Tissue biopsy, again it is difficult, but again it is also coming up with a peripheral blood mononuclear cell. So again this is something which can be used for electron transport chain also. So I feel this looks impressive for me because I will be able to look at the ETC as well as the oxidative phosphorylation. Again, cell lines I already discussed, it is difficult. So now I am coming with a new problem. I am telling that I am having limitation in funding, right? So I am asking it which laboratory test I can use for both electron transport chain oxidative phosphorylation, and these tests should be less expensive and the test should use peripheral blood mononuclear. So I am very precise thing I am telling. First thing I am telling is the funding issue I am having. Second, I am asking it the test for electron transport and oxidative phosphorylation, which should be less expensive, and I should be able to use peripheral blood mononuclear cells for the tests. So again, it is telling that because of your budget constraints, cost-effective method, which can use peripheral blood mononuclear cells, it's coming out with spectrophotometric assays 1, which is relatively inexpensive. It is also suggesting me a coenzyme Q redox state and mitochondrial membrane potential. 3 things it is suggesting from electron transport chain. And for oxidative phosphorylation, it is giving me suggestions on ATP production assays by looking exactly at the ATP levels. Then it is suggesting me to look at the oxygen consumption rate, a very frequent thing which is used. And it is also suggesting the most important analyzer, which is the CHOS XF analyzer. And of course, mitochondrial respirometry. So all these can be done with peripheral blood mononuclear cells, right? So it's given me many things. So just to summarize what it has given me, I have just literally started from scratch. It gave me a hypothesis of 3 hypotheses from which I selected 1. Then from there I wanted to look at the electron transport chain and oxidative phosphorylation. It gave me the set of possible tests and set of possible samples, and I chose ETC and oxidative phosphorylation and peripheral blood mononucleosides, and I wanted tests which are less expensive, and was able to give it. So the general idea it has given me. So now I'm ready with a research concept with some idea about what I can go, and for each of the aspect which I've so far looked at, I can actually ask more information from it, and I will be able to get more and more information. And of course I have to ultimately go and read those research articles to build my research, but it's a very good beginning point for me to take it and show to my PhD guide and say that I have a concept in mind. And please be transparent to your guide when you use AI. It's very important that we use AI and we disclose. We have not stolen anything. We have just used it as a virtual PhD guide, and we've gotten information. So this is just a basic outline. Be tuned in for more specific methods of prompt engineering, how to give prompts and how to give information, get information from the AI model, COPILOT. And the same is applicable for the ChatGPT 4, which is available for a cost when you pay about 2,000 a month, or 3.5 is available free. For all these research methods, I think 3.5 is as good as 4, so you can use it for your research methods.